start our recording. All right, so now we're recording. So, all right, y'all, I'm going to go ahead and run through this PowerPoint. Along the way, please ask some questions. It's probably going to take me like 20, 30 minutes to get through all this, depending on if we have questions or not. Okay, so I always start this lecture out by just talking about generally why we hypothesize sexual reproduction exists in the first place. Because organisms like bacteria, they just divide, and that's how they replicate themselves. Whereas we produce gametes, which are haploid cells. So we have 23 chromosomes in each. Sperm and egg come together and create a single-celled zygote, where we have the recombination of our full set of 46 chromosomes. Now, we hypothesize that the reason sexual reproduction exists is so that we can increase our genetic diversity in our population, which helps us compete with things like pathogens, bacteria, fungi that replicate at a much faster rate. So that's just like a general why we have sex. It's pretty interesting. If you want to talk about that, we can have an email conversation at a later date. So, generally speaking, we inherit 2 chromosome 23s, one from our mother, one from our father. We can define chromosomal females as having 2 X chromosomes and chromosomal males as having XY chromosomes. Now, on this figure, it says male and female, whereas I'm intentionally choosing to use chromosomal female and chromosomal male to be as inclusive as possible. For instance, I've read an article about a woman who had an X and a Y chromosome and had a healthy birth. She was able to do this because her cells did not respond to the Y chromosome. So although most of her cells had an X and a Y, she was essentially XO as a female and gave birth. So when we consider inclusive language, how we define male and female can vary depending on your definition of male or female. So if we're talking about chromosomal inheritance, I will use the language of chromosomal male and chromosomal female. All right, this is not included in your terms list for the semester, but anyone working in healthcare should be aware that not everyone has textbook binary genitalia that you'll see in the images that I'm going to present throughout this lecture. So, intersex individuals are, I've been told, just as common in the population as people with red hair. And while a dental hygienist might not see genitalia such as this, if you're a nurse or a doctor, physical therapist, you may see something like this. I don't know about physical therapy, but if you're a nurse, you should expect throughout the duration of your career to see intersex genitalia. And all that I ask is that you be respectful and not view it as abnormal or nasty or anything negative like that. These are people with feelings and deserve respect just like everyone else. All right, I pulled this table here from Wikipedia, and this just shows that developmentally, males and females have homologous structures. So for instance, the testes and the ovaries, they're both gonads. They produce the gametes that will recombine into a diploid zygote. Let's take a look at the scrotum. So in men, the scrotum is homologous to the labia majora of females. The penis is homologous to the clitoris. And we'll talk about several of those. And so I really like this image because it does show the homologous nature of the clitoris and the penis. Typically, the representation is shown only in the vulva of the clitoris, whereas this elongated region actually straddles the vaginal canal. And as with the penis, this rooted structure goes into the abdominal region and provides, again, like a rooted structure so that the penis can become erect and penetrate whichever orifice that individual chooses to place it in. Okay, this is a pretty important slide, so hang with me here and let me know if you have any questions. Okay, so the male reproductive system is what we're going to describe as closed. And what that means is all of the cells and the fluids are going to stay contained within a set of tubes that will be released through the penis. So sperm cells are coming from the testes. We have fluids from 3 different regions that we'll get into, but everything is contained within a closed set of tubes. So you can think of this as turning on your garden hose. You turn on the spigot, water flows through the hose and out the end of it. That is a closed system. When we consider the female reproductive system, this is an open system. So take a look here where my cursor is. The ovary will release an egg into the abdominal cavity, and then the fallopian tube has these finger-like appendages called fimbriae. And these are going to kind of undulate and wave the egg and bring it into the fallopian tube. This is open though. So eggs are released and then grabbed by the fallopian tube. This is not directly attached the way that the vas deferens is attached to the testes. So this is open, whereas male repro is closed. Does anybody have any questions about this slide? Okay, cool. So let's keep rolling. Okay, generally speaking, we associate testosterone with maleness. So this is going to be secondary sexual characteristics, things like increased facial hair, for example, potentially deepness of voice. Whereas female secondary sexual characteristics like breast size or hip width can be regulated through estrogen and progesterone. Now, these also play other roles, but they play a role in sexual secondary characteristics as well. However, I include these images in the bottom just to show that even within a female, within a male, we can interconvert and produce one hormone from another through the use of enzymes. All right, so now we're going to go through the male reproductive system. I would generally go through females first. However, the male system is more straightforward, so we're going to start with the easier content and then move into females as we wrap up our discussion. So, starting with the site of sperm production. So, sperm are the haploid cells. Again, haploid means we've gone through the process of meiosis, not mitosis, but meiosis, M-E-I, and we've created 23 chromosome cells, and they are produced in the seminiferous tubules. From the seminiferous tubules, we also have testosterone production, and the sperm cells will mature and wait in the epididymis. So when I say wait, I mean they're stored and being ready for ejaculation. So they'll be in the epididymis, which is superficial to the testes, and from there, during ejaculation, the sperm will leave and come out through the ductus deferens. You've probably heard this be called the vas deferens, V-A-S deferens. These are synonyms. So whether you call it the ductus deferens or the vas deferens, they're the same thing. So that is where sperm cells will travel through. Notice that we have blood vessels and nerves here as well, and they are wrapped, protected in the spermatic cord. So each testis has a spermatic cord that will wrap around its blood vessels, nerves, and vas deferens. All right, so now let's talk about what semen is. Semen is not a synonym with sperm. Semen is a fluid that contains sperm as well as other components. So we have sperm coming from the testes, stored in the epididymis. It's going to travel through the deferens. Then we will reach this ampulla, which will join with a seminal vesicle, shown here in yellow. Here, this is a frontal cross-section to show the opening of the seminal vesicle as it meets with the ampulla coming off of the vas deferens. So what's going to come out of the seminal vesicle? We have an alkaline or a basic fluid, and this is the majority of the fluid found in semen. So it's alkaline. It's going to help counteract the acidity of the vaginal canal, because we want our sperm to survive so that they can go and fertilize an egg. So you'll notice down here, once we get past the seminal vesicle, so this is the point where we have what's called an ejaculatory duct. This is the fusion, this Y shape, this fork, where the ampulla from the vas deferens will meet the seminal vesicle. This is the ejaculatory duct. From there, we're going to have the right and left sides meet at the prostate gland. The prostate gland also produces a large component of the fluid found in semen, about 30%. This is acidic. It contains citrate, which is a sugar, as well as some enzymes. They're going to help keep the sperm alive and moving so that they can reach and, again, fertilize an egg. So we have fluid from the seminal vesicle. Then the two seminal vesicles meet in the prostate. Prostate, again, secretes fluid. We also have in the abdominal floor two, one on the right, one on the left side, bulbourethral glands. So bulb, it's like a small bulb shape, and then it meets with the urethra. So bulbourethral gland. It's a gland, it secretes a fluid. This fluid is also alkaline to neutralize the acidity, and it's going to provide some lubrication, and this is commonly referred to as pre-ejaculatory fluid. You'll notice that this is the closest gland to the outside of the penis. It is the closest gland to the urethra. Therefore, this is the first component that will be released during an ejaculation. So we have three types of fluids also containing the sperm cells. Keep in mind, sperm is not the same as semen. Here we have a sagittal view of what we just talked about. So showing the seminal vesicle meeting with the vas deferens here. So down here we have our ejaculatory duct meeting with the urethra coming from the bladder. So the bladder has the trigone, remember, where the two ureters, so this is a ureter on the right side, there'll be a ureter on the left side, and then that third part of the trigone is the urethra. That's going to be an inferior feature. So here's the urethra right here where my cursor is. That meets the ejaculatory ducts on the right and left sides. So we have like a three-way Y fork shape meeting in the prostate gland. After that 3Y fork, then all of those channels meet and leave through the abdominal floor as the urethra. So the bulbourethral glands also secrete their fluids into the urethra, and from there it is one way out. So urine as well as ejaculant will leave through the urethra in penis. So does anybody have questions about the fluids or the branching structures of the ducts in this closed male reproductive system? All right. There are two muscles that you should be aware of as far as the male reproductive anatomy, and both of them are going to serve the same function. Whenever the external environment is cold, these muscles will contract and bring the testes closer to the body, and that's going to help protect the sperm. We need to do this because the testes are external as opposed to the ovaries, which are internal in people with female reproductive structures. So let's start with the cremaster muscles. The cremaster muscles are attached directly to the testes, and these will bring the testes closer to the abdomen. These are continuous with the internal oblique muscles, and depending upon which source you look at, you could see that they are involuntary or voluntary to some degree. You can think of this as kind of how the diaphragm contracts involuntarily, but you can also have some voluntary control over the diaphragm should you choose. I had a student one time show me a Snapchat or an Instagram, some sort of social media, where her friend, who is a man, was retracting his testes very quickly and very voluntarily. So should you have these structures, I recommend you give it a try on your own. Try to contract your internal obliques. So really contract your abdominal muscles, and you may see testes retract. If you don't have these structures, talk to your male friends, talk to your boyfriends, your partners, whatever it is. See what you can learn. As for the dartos muscles are going to line the scrotum. So that's this pink line lining the scrotum. These will also contract and make the scrotum shrink and, again, bring the testes closer to the body when it's cold. As for how an erection takes place, men have 3 erectile tissues. There are 2 structures on the right and left side that are superior structures called corpora cavernosa. And then the corpus spongiosum is going to line the urethra. So the corpus spongiosum is going to keep the urethra erect so that the ejaculatory fluids as well as the sperm can leave the urethra functionally, whereas the corpora cavernosa are the larger structures that provide the more rigid structure of an erection. All right, now let's roll into female reproductive system. Be careful which images you look at. Notice this image shows the system essentially as a closed system, but as we said, the ovaries are not directly attached to the fallopian tubes. You'll also notice that this says oviduct, and oviduct is a fallopian tube. So whether you choose to use oviduct, fallopian tube, uterine tube, they are all synonyms and they are all fair game for your second practical. This image is also useful to show why pregnant people tend to have increased need to urinate frequently. So as the child develops in the uterus, it is going to press on the bladder, which is going to be inferior to the uterus, and that can make someone need to urinate more frequently. All right, as for external female genitalia, we're going to refer to this as the vulva. This is not on your terms list, but I think anybody in an anatomy class in college should learn this just in case you haven't before. So, things on your terms list are the clitoris. Again, I mentioned that we can only see the glands from the view of the vulva, which is why I included that image earlier to show the entirety of the clitoris. The external labia are the labia majora, so the major labia, and these are going to have hair growing on them, whereas the labia minora will be more medial and they will not have hair growing on them. The vagina is a canal that will eventually lead to the cervix, And then the cervix is going to be our cutoff between the vagina and the uterus, and we'll talk about that in an upcoming slide. The perineum is present in both men and women, and this is the region between the genitals and the anus. The image over here on the left that I'm showing, I've included as a sort of almost like social justice for women. So referring back to that homologous structure slide, depending on which book you look in, you may or may not see Skene's glands as well as Bartholin's glands. Whenever I bring this up in class, I ask, have your gynecologist ever told you about these two glands? And more often than not, women say no, and men have also never heard of these. So for your own sake, I'll go ahead and tell you that the Skene's glands are the homologous structure to the male prostate, whereas the Bartholin's glands are the homologous structure to the male bulbourethral glands. Both of these are glands, therefore they will secrete fluids. So again, have you not heard of these? I recommend that you do a little bit of independent research and learn about your own body or your partner's bodies or the other half of the human population's body should you have male reproductive structures. Okay, as I was learning more and more about what is present in the literature about female reproductive anatomy, I noticed that there's a lot of people who are essentially arguing over what is present in female reproductive anatomy. And this is usually a fun little topic that we discuss in class, but you can review this on your own time should you want, and we can talk about this at a later date through email or through some sort of Skype message if you're interested in this discussion. And I included this just to show the complete structure of the clitoris alongside the other reproductive structures that we just talked about. And then this would be the time in class where I would pull out a 3D printed plastic model of a full clitoris and place it within a female pelvis to show that the clitoris actually does straddle the vaginal canal. All right, now let's take a look at a more textbook-esque image of female reproductive anatomy. So we have 3 general layers of the uterus to be concerned with. The deepest layer is called the endometrium, and this is what is shed on a monthly basis. The muscular layer is the myometrium. Keep in mind that myo means muscle. And then the external, most superficial layer is the perimetrium. Peri like perimeter. So this is the outside. So those are the 3 layers of the uterus. Let's remember that this is an open system, and that's really well provided in this image. So the ovary will release an egg once a month. Generally, I've been told and read that ovaries will switch. So this month, an egg will release from the left side, whereas the next month, an egg will release from the right side. So an egg is released, and then the fallopian tube has these fimbriae, these finger-like appendages that are going to undulate and bring the egg into this first region called an infundibulum. So infundibulum, ampulla, and isthmus, those are all regions of the uterine tube, which again is the same as an oviduct, which is the same as a fallopian tube. Those are all synonyms. So the egg will travel through and stay in this fallopian tube, which is where it's generally fertilized by sperm. So the sperm will travel through the uterus, up through the isthmus, and generally meet in this ampulla region or in the isthmus region of the fallopian tube. After fertilization takes place, then the zygote, which is the single cell, will divide. Once it divides into 2 cells, then we call it an embryo. The embryo will literally roll into the uterus and then hopefully implant into the uterine wall. there can be big problems if the embryo implants in the fallopian tube. If this happens, then the ball of cells that we call an embryo will eventually get so big that it causes a rupture in the fallopian tube, and the embryo will not survive that. I've never seen a case where it does survive. Again, with biology, there's always an exception to the rule, so maybe it's happened before. Generally speaking, you're going to have a miscarriage, and the mother will experience a hemorrhage. So that's very dangerous for both the mother and the potential offspring. But again, keep in mind, fertilization almost always takes place in the oviduct, but implantation occurs in the uterus. So from the uterus, we're going to travel more inferior until we get to the cervix. The cervix is the cutoff between the uterus and the vagina. The vagina is lined with rugae, which allow it to increase in surface area. And then we will leave into the external region, which again we refer to as the vulva. You do have 3 ligaments that you should be aware of on your terms list for the female reproductive anatomy. We have the ovarian ligament. The ovarian ligament will attach the uterus to the ovaries, or the ovaries to the uterus, however you want to learn that. So the ovaries, keeping in mind that they're not attached to these fallopian tubes, they're going to attach to the external structure of the uterus. Keep it intact and where it needs to be. We have the broad ligament, which is going to be the most superficial. It's going to cover the top of the uterus and essentially be like a sheet on top of all of these reproductive structures. And then we have a round ligament, which again will attach to the outside of the uterus, and then it will travel down and attach to the labia majora. This is an image that Dr. Mansfield-Jones included in my prep notes for today's lecture. She has this suspensory ligament included in this sagittal female reproductive anatomy model. From my memory, the suspensory ligament is not on your terms list. But you can see right here, this broad ligament, you can see where it's ending. So anything that is superficial to the uterus, if you're asked for a ligament, will be the broad ligament. Here you can see the ovarian ligament. So here's an ovary. Here is this number 16 is the ovarian ligament. So this would be the fimbriae with the fallopian tube traveling to the uterus. And then here A is showing the round ligament, again showing attached to the uterus, and then it will attach to the labia majora. I've included this embedded video, so if you are interested, you can type this title up here into YouTube. There's a really cool video that you can watch on your own time where it shows an egg being released from an ovary in its follicle and being grabbed by the fimbriae. I definitely recommend you all watch this. It's really fascinating. All right, women, whenever they become pregnant, will begin lactation. So mammary glands are present in mammals, so they will produce milk, and that milk is both food as well as an immune booster for the offspring. So any sort of antibodies that the mother is producing through her exposures will be transmitted through the milk to the young. And these are modified sweat glands. All right, so let's see, we spent about 30 minutes or so going through that, and we have gotten through everything. So at this point, we can ask questions, or if you all want any sort of a review, you can ask me for it, and I will talk your hearts out. I will be present in this webcam for another hour. So I'd like to hear from you all. What would you like to discuss, or do you have any questions at this point? Hang on one second. Let me close my window. Somebody's got a leaf blower outside. Okay, am I going to collect homework for this? No, I am not going to collect homework for this. So you should do it. You should read the article. The article is about an ectopic pregnancy, and I would expect her to ask one question about the ectopic pregnancy, just to make sure that you read the article. But I will not collect the homework for points.
Hey, is it okay if we do like a review over what's going to be on the practical?
Yeah, of course. So let me run through the topics real quick, and then as I go through the topics, just think like, you know, I'm not so sharp on that. What could I use a review of? And then we can spend as much time as you want over this next hour going through anything. I can pull up the biopack and share my screen. So let's just start at the top and work from there. All right, so here's what's on the exam. You should know heart anatomy, so all the chambers, as well as systemic blood vessels. You should understand blood flow through the body, as well as through the heart, so systemic and coronary circulation. So that's heart anatomy, and then heart physiology is in the EKG. So EKG, ECG, electrocardiography. Then our next topic was pulmonary anatomy and physiology. So that has to do with the pulmonary system. So everything from the nasal passageway going into the pharynx, the trachea, the bronchi, the lungs, etc. So the difference between the conducting zone, the respiratory zone. And then we also had the pulmonary volume biopack, where we measured tidal volume, breathing rate, alveolar minute volume, the forced expiration volume in 1 second, total lung capacity, right? So we had all those different volumes that we talked about. So it's heart, pulmonary, and then we did the aerobic exercise biopack over this past week, as well as the digestive system and the excretory system, or the urinary system. So that includes the kidneys, the ureter, bladder, urethra, difference between men and women, similarities between men and women, and then into today's content about sexual reproductive anatomy. So those are all the topics. Is there anything that you all would like to start out with any sort of a review over?
Could you go over like the pulmonary system again? Like not the whole thing, but like more so the EKG part.
Okay. So Naya, I do
Yeah.
have one question just to kind of figure out what you want to go over. So you said pulmonary, but you wanted to go over the EKG. So the pulmonary was the lung volumes, and then the EKG was the biopack lab before that. So
I mean, not the EKG, the biopack. That's what I meant to say, not EKG.
okay.
Sorry.
Don't worry about it. So do you want me to pull up the biopack and like show how to highlight stuff and all that?
Yes.
Okay, hang on 1 second, all right?
Okay.
And let's do here, and then share content, share application window. Okay, hang on. I'm gonna go grab my water bottle real quick. Hang on. Okay. So going through this file that you all worked through, what we see is that we can change our tidal volume. Keep in mind that tidal volume is just a repeated breathing pattern. So we can breathe faster or slower. We can breathe deeper or more shallowly. So for these first series of breaths, we have normal breathing pattern. So what that means is that we have a breathing rate that is fairly constant, and we also have a volume that is fairly constant with inhales and exhales. And then intentionally, we can choose to breathe shallowly. If we breathe shallowly, we might expect that we're going to breathe more quickly, and this is something like panting. So if you're breathing shallowly, you're getting less volume in and out, therefore you might breathe more frequently. Then we return back to normal breathing as we see the tidal volume increase back to a higher number, a higher range of volume. And then we're going to go into a deep breathing period. So with deep breathing, we see that the tidal volume increases with the inhale as with the exhale. And generally, if we're deep breathing, this is either going to be during an aerobic state or maybe during some sort of like meditative, tranquil state. During exercise, we expect our breathing rate to go up. We also expect our tidal volume to go up. Both of those are going to cause an increase in the AMV, which is the alveolar minute volume. And we can talk about that here in a minute. As with our deep meditative or tranquil breathing, this is intentionally deep and probably more elongated each breath, which will cause a slower breathing rate. Keep in mind, breathing rate is measured in breaths per minute. So what we've done is we've gone from normal to shallow, back to normal, back to deep, or too deep, and then back to normal. And then now here, we have a repeated series of breaths so that we can measure FEV1. Before we do that, I want to go and show you how to measure tidal volume. So we're just going to do one thing at a time. So let me zoom in, zoom in, zoom in, auto scale. Okay, so we are looking at normal breathing right here. So let's talk about our different tools. So we want channel 2, we want a peak to peak, we want delta, and we also want delta T. All right, so peak to peak, delta, and delta T are the only tools that we have used this whole semester, okay? Now, we can measure tidal volume 2 different ways depending on which tool you choose to use. So let's start with this method. Now, if I start at the very top of this peak, so this is after an inhale, and now we are beginning the exhale phase. So I'm going to highlight half of a breath. This half of a breath is an exhale. Notice that my peak to peak and my delta are the exact same quantitatively speaking. Here we have a negative, but you can ignore that negative because when you look on your scale on the right, you see that everything is positive because we're measuring volume. So the reason this is a negative value is because BioPAC is going to calculate end minus start. That is by definition what delta is. So if we have this value, which is like about 2 minus roughly 3.4-ish, we have 2 minus 3.4 gives us negative 1.4. If I had started my highlight here and ended here, then I'd have 3.4 minus 2 equals positive 1.4. Okay? So if you see a negative value here, ignore the negative sign because everything is positive when we talk about volume. Volume is represented in liters on this y-axis. So if you have half of a breath highlight, your peak to peak and your delta should be the exact same number. But let's say that you start with an inhale. So this is increasing volume. So that is one inhale. So I'm going to have an inhale and an exhale. So if I highlight one entire breath, you'll notice that my delta value is approximately 0 because it's saying, all right, well, this is where you ended, that's where you started, and then if we go to our y-axis, those are essentially the exact same value. So delta equals 0. But your peak to peak considers the minimum and the maximum value. So peak to peak, by definition, is maximum minus minimum. Delta is end minus start. So if you have one entire breath highlight, you should expect your delta value to be 0, but your peak to peak to have accurate representative data.
Okay, thank you. That makes more sense now.
Great. That it can be complicated, but that is one big hurdle that we're just going to go ahead and get over. What is delta? What is peak to peak? So what I recommend you do is on an exam, on a quiz, or I guess we don't have any more quizzes. So on your exam, or I guess you don't get a paper exam, as you're studying for your exam, go ahead and write this stuff down. So peak to peak equals max minus min. Delta equals end minus start. Both of these, peak to peak and the delta, only consider the y-axis. Delta t is end time minus start time. So it's the exact same thing conceptually as delta, but it's only going to consider the x-axis, which will always and only be time. So what delta t does is, let's say I start here at about 140 and go to about 142. What this is saying is 142 minus 140 equals 2 seconds. So these are the only tools, and if you can understand why delta t works, that should make understanding how delta works. Peak to peak has the name in it. What is the maximum peak? What is the minimum peak? Subtract one from the other, you're going to get a value. So that is, you are so welcome. All right, so that is how we're going to measure our tidal volume. And keep in mind that tidal volume will decrease with shallow breathing and increase with deep breathing. As for the breathing rate, we can highlight 1, 2, 3, even 4 breaths, right? So you're going to start and end at the same place. So if you start at the transition between an inhale and an exhale, you're going to want to stop at that place as well. Or you could start at the bottom and just make sure that you have a complete number of breaths. What you don't want to do is something like this, where you have 1, 2.5 breaths. Now, you can still calculate your breathing rate this way, but you get a more accurate measurement if you include a complete number of breaths. So what we're going to do is, just like heart rate, number of cycles over delta t times 60. So in this case, I'm going to have 1, 2, 3 breaths divided by 10.78 seconds, multiply that by 60 seconds over 1 minute, and I'll get my breathing rate. So questions about how to calculate tidal volume or breathing rate, or are we good?
I'm good with those now.
Cool. Let's talk about the other ones. Naya, how do you feel about ERV and the IRV?
I can do those pretty well. I think the main one was really just like the tidal volume and I think vital capacity.
Okay, gotcha. So do you recall what the vital capacity is? Like by definition, do you remember?
I know it's like the total amount of air that goes in and out of your lungs or something like that.
Right, it is. So it's going to be the amount of gas that an individual can inhale and exhale. So it's the complete volume that you're able to take in and let out. But after you exhale all the way, you'll notice down here that my max exhale bottoms out at 1 liter. So the BioPAC can't measure this 1 liter, but we assume it for adults. This is called the residual volume. So the residual volume is a volume of gas that is present in the pulmonary system after a full, complete exhale. So think about it this way. If you were to take a vacuum, like a household vacuum, and suck all the air out of something, you're going to end up with a shriveled, imagine a balloon, right? A balloon at rest, if it's not tied, like you just open up a balloon out of a bag, there's some gas on the inside of it. But if you connect that to a vacuum, you'll suck all of the air out of it. Your nasal passageways, your mouth have gas. Your trachea has gas. Your entire pulmonary system would have to collapse on itself for this to be 0, and you wouldn't survive that. So think like your tracheal cartilage, for example. Or I guess that's not a good example. Let's see. Yeah, sure it is. Tracheal cartilage is going to keep the trachea from collapsing on itself. So that's a protective structure to maintain a residual volume. So If we were to do a quantitative assessment of this, we can say that the total lung capacity equals the vital capacity plus the residual volume. So the vital capacity we can measure using this highlight region, which again, if I start at a maximum and end at a minimum, should be the exact same value with a peak-to-peak and a delta measurement, because I'm starting at a maximum, I'm ending at a minimum. So max minus min is end minus start. So by definition, peak-to-peak equals delta for vital capacity. So what you should expect to see on your practical next week is a screenshot, maybe with, for example, if you were asked which functional volume is displayed in the highlight region shown in this screenshot, you would say this is the vital capacity. And then if it said what and provide its measurement, then you would look up in either of these two boxes. You might be given the peak-to-peak, you might be given the delta, you might be given both. You might be given them just like this, where you have one that's negative but one that's positive, but the number is the same. And you should know that you should ignore the negative sign because all volumes are positive. So you should know what is highlight and then also which box to choose, keeping in mind that you only have peak-to-peak, delta, and delta T. Delta T will never measure a volume. It will only measure time, and you can see that in seconds with the units here. So was there anything else about vital capacity that you were wondering?
No, that was pretty much it. I think I was just like getting it confused with one of the other measurements.
Okay, was it the FEV1 by chance?
Yeah, it might have been that one. Because I think I
Okay.
might have switched it on while I was taking the quiz.
Okay, so let me show you FEV1. So with FEV1, we're going to start after, so here we have normal breaths on the left and normal breaths on the right. So what we're doing is we're going to inhale, increase volume, exhale, decrease volume, and then do our full inhale. So this is the maximum inhale, and the difference between the normal inhale and the max inhale is shown in the delta value. So that is the IRV, right? So yeah, so from that max inhale, we're going to go down as we exhale. We're going to continue until delta T equals 1.0 seconds. We're going to highlight 1 second of a forced exhale. Then we're going to measure that volume using either peak-to-peak or delta. Notice that they're the exact same because, again, that's my max, that's my min. That's my start, that's my end. They're the same value. So we get a volume here. So this volume is 3.24, I'm just going to write 3.25 liters. So that is the FEV1, the forced expiration volume in 1 second. So that's where you forcefully exhale as hard as you can. How much volume can you let out in 1 second? This is useful in a clinical setting if we consider it relative to the vital capacity. So if we have a vital capacity of 4.7 liters, what we can do is we can say the FEV1 was 3.25, so 3.25 divided by 4.7 gives us a ratio of FEV1 relative to the vital capacity. So that's useful because if you can let out all of, let's say that this delta T was 1 second. If you can let out all of your vital capacity in 1 second, that indicates that the airway is probably not obstructed or restricted. Those are different. Obstruction comes from things like mucus or tar. Maybe I don't know about coal miners. I don't know if they have any sort of like coal deposits that obstruct their airways. That's different from restriction, which has to do with an allergic response. So if somebody's really sensitive to some sort of environmental exposure, their airway can restrict. So things like pollen, or let's say someone goes into anaphylactic shock. Their airways can constrict and make it hard to breathe, and they would have a smaller relative FEV1 to their vital capacity. So the same way that if you can let out all of your vital capacity in 1 second, that's a healthy, unobstructed, unrestricted airway. If you cannot let out much of your vital capacity in 1 second, that indicates that you could have some obstruction, you could have some restriction taking place.
Okay, that makes more sense now.
Right. So this might be something that you all do every day if you're working in a hospital or in a nursing home. All right, so we have Josh and Seth. So we have talked about all that. Do you have anything to talk about?
I think I'm good.
Okay. Naya, is there anything else you want to talk about? Anything at all?
I think that was pretty much all. Like the
Okay.
only big thing that I was really stuck on.
Okay, sounds good. All right. So here's what I'm going to say is I will be, wanted to discuss blood pressure with aerobic exercise. I think I understand it, but just need a recap. Okay. Blood pressure with aerobic exercise. All right, so as we start our aerobic exercise, we're going to have the sympathetic nervous system become activated. So the sympathetic nervous system is going to do several things. It's going to make the heart rate increase, so the heart's going to beat faster. It's going to make breathing rate increase. And we're also going to start, let's see, we have the vascular shunt, which has to do with vasoconstriction and vasodilation. Excuse me 1 sec. So with aerobic exercise, we want blood to go to our skeletal muscles, right? We want blood to restrict its flow to areas that are not the skeletal muscles. So things like the bladder, the gastrointestinal tract, the kidneys. We're going to limit blood flow to there because we only have so much blood available. The typical adult has about 5 liters of blood. So if we want oxygen to get to our muscles, we want CO2 to get out of our muscles, we're going to dilate those blood vessels, and we're going to constrict blood vessels elsewhere. If heart rate increases, if stroke volume increases, we're going to have cardiac output increase. We're also going to see blood pressure increase. So we expect blood pressure to go up with exercise. Now, that gets us into the firefighter article about expectations versus what's wrong with the firefighter. So, can anybody recall what the firefighters' issue ultimately was? Okay, so the firefighter could not vasoconstrict when he needed to have exertion. Okay, so that is part of it. So let's talk about that. So what's going on is that the firefighter had sympathetic autonomic failure. So keep in mind that the autonomic nervous system is not under voluntary control. So autonomic, it happens on its own, right? So sympathetic is the increased heart rate, increased breathing rate side of the autonomic nervous system, and the parasympathetic is the slow down heart rate, slow down breathing rate side of the autonomic nervous system. Since the firefighter had a functional parasympathetic nervous system, but a dysfunctional sympathetic nervous system, what did not happen was he did not have vasoconstriction to the blood vessels serving outside of the skeletal muscles. Therefore, his blood pressure didn't go up as high as we expect it to because he had remaining vasodilation where they should have constricted if his sympathetic nervous system was functional. So his heart rate doesn't increase very much with exercise. His blood pressure doesn't increase much with exercise. It does a little bit, but not much. Not as much as we expect it to. So do you all recall what keeps the blood pressure up enough during exercise to prevent him from fainting during exercise? Because he doesn't faint until he quits exercise. So do you all remember what kept his blood pressure up? Increased cardiac output. Yes, so he did have, again, like a slight increase in cardiac output. So keep in mind that cardiac output equals heart rate times stroke volume. The sympathetic nervous system is going to cause an increase in heart rate and stroke volume. But since his wasn't functioning well, or not functioning well, his sympathetic system wasn't functioning at all, so far as we could tell. He had a slight increase in his cardiac output only because he was able to maintain his venous return, because he had a leg muscle pump. Okay, so the leg muscle pump is mentioned in the article. And what this means is that during exercise, so he's standing up, he's using his leg muscles. When the leg muscles contract, they imagine this is a blood vessel and this is a leg muscle. So as the muscle contracts, it squeezes on the vein in the leg, and that's going to squeeze the blood up and increase venous return into that inferior vena cava. Because, again, literally the leg muscles contracting squeeze and increase pressure to push up against gravity and increase venous return. That increased venous return is going to cause an increase in the stroke volume, which will increase our cardiac output. So after he stops exercising, then his leg muscles are no longer contracting. Therefore, he's no longer increasing that venous return, and his blood pressure plummets. That's why he passes out. Rather, that's why he passes out after exercise and not during exercise. So the things to consider for the firefighter, there are, I guess, like three big concepts. Sympathetic autonomic nervous failure. The leg muscle pump increases venous return, and when he stops exercising, his blood pressure plummets because that leg muscle pump is no longer functional. Therefore, he passes out after exercise. So that's the kind of takeaway message for that whole article. And you should expect to see at least a question or two about the firefighter. All right, so we have recapped over our lung volumes. We've talked about blood pressure with aerobic exercise. We've talked about the fainting firefighter. What else do we have questions about? She has a lot of questions about it in the last 2 weeks. That article is relevant to a lot of things we've talked about. Yep, that's right. She loves this article. We've used it for several weeks. You've been asked plenty of questions about it. Therefore, she thinks it's very important, and you should expect to see it on the practical. And yeah, it is really important because it ties in the nervous system with the cardiovascular system, with the nervous system, with a discussion of hemodynamics. So yes, this firefighter is a very inclusive type of article that you should expect to see. So it's like 2 or 3 pages. Read over it again. Look at the figures. I've never seen her provide a figure from the article, but with the kind of open-ended nature of the exam next week, you could see an image from that article. So let me just mention that. So for those that weren't here earlier, I have not been in communication with Dr. Mansfield-Jones for the past several weeks, just because everyone's isolated at home and kind of working like crazy with this transition to online classes. But I'm meeting with her tomorrow. So we're going to meet and talk about the practical. It has not been written. We don't even know what the format is going to be like. I don't know if you should expect to see model images, but there are model photos available in the PowerPoints and in your Blackboard Week folders. So definitely review those for your own sake and also just in preparation for the exam. I don't know if we're going to have images like on a quiz or images out of the textbook. I highly doubt we're going to have textbook images. She'll probably provide you with either a model or some sort of a novel photo like you see on the quizzes from early 19th century, late 18th, or early 1900s, late 1800s, because those are open source for use. So is there anything that you all want to have me run by Dr. Mansfield-Jones when I meet with her tomorrow? If there's anything that you want to know about this class, I can ask her directly, and I will post an announcement with some information after I talk with her tomorrow.
Do you know if there will be like a practice exam like last time?
I have that saved on my computer, and I will make a note of that right now. Post practical number 2 PowerPoint. So what I'll do is I'll post that as soon as our session ends here. I'll put it in course content, so it'll be below all the stuff that I post. So don't go into course information or laboratory information. Just go course content, and then I'll put it at the bottom down there next to all the other colored links that I've provided. And so that's from an old exam. I don't recall if it has brain and cranial nerve questions on it. If it does, be aware that brain and cranial nerves are not on this practical. But the PowerPoint that I was using for this practice practical number 2 may or may not include brain and cranial nerves because it used to be on the second practical, but it is no longer on there. So I will post that again here in the next 20, 30 minutes, and then I'll leave an announcement for you. And that is just like the first one where it's timed with 2 minutes per slide. So if you open up the slideshow, every 2 minutes you'll get a new slide with an image and 2 questions. So it'll be things like, what is structure A, and maybe what is its function, right? So as you're studying, you definitely want to have all your terms lists out and make sure that you know how to identify each anatomical structure and then what is its function. But yes, I will, in the very near future, in the next half hour, have a practice practical for you.
Nice.
For sure. And I think that I included a short answer section in there. I don't recall. But I do know that I definitely have the 23 stations that I'll provide. Okay. The last homework was worth 104 points. Will that translate into the regular amount of points for homework? Yes. So the 104 points that you're talking about will be converted into a 12-point assessment for that. So the reviewing your knowledge is worth 12 points, and the patient connection article response is worth 8 points. So out of the 104, I will take all of your correct answers and divide that by 104, and then multiply that fraction by 12. And then that will be your grade on top of 8 potential points for the patient connection. So therefore, your homework 5 will be just like the other 4 homeworks out of 20 total points. And I'll have that in a separate gradebook column. Okay, yeah, I've been told about this issue as well. So the comment is that some answers were input as correct, but were provided to you as a wrong answer. So the way that this works is we have to program every potential student response as correct. And if you provide something that is an abbreviation, or maybe like you put an S and made it plural, we don't program that as a potential answer, it will mark it wrong. So what I'm going to have to do is I'm going to have to go in and edit the document such that if you put capillary or capillaries as plural, that both of those are correct responses. So that's going to just take me going in and manually adjusting some stuff. And then once I do that, Blackboard regrades everything automatically, and it will update your grade. So I'll make sure to do all of that before I create that new column, which will be homework 5 out of 20 points. But I know that like if you put GI tract as opposed to gastrointestinal tract, those are 2 different answers. So if Blackboard isn't programmed to accept both of those, then one or the other would be wrong. But for our purposes, they are both correct. So you will get credit for those. Right, it is, and I'm glad that you're concerned with your grades. It is definitely appreciated. All right, so we have 10 minutes left of this online virtual classroom. So do you all have anything you want to talk about? If you all feel confident with everything we've talked about and you don't have any questions, you can feel free to go ahead and leave this room. I'll be here for the next 10 minutes. So if you want to go ahead and dip out and then come back in, if you do have a question, that's fine. You don't need to stay here for the next 10 minutes. And then as soon as we're done with this session, I'm the recording and it'll be available on Blackboard. And then I'll also post it to my YouTube channel with a link in the announcement that you'll see tomorrow. Cool. Josh, I appreciate you being in here. Just shoot me an email if you have any questions about anything. Like I said, I'll be here for the next 10 minutes if you do need to come back and ask anything, all right? I hope you have a good week as well, and I'll see you soon.